Okay, this is part 3 of chapter 7, but the good news, we're on section 7.2. I think we can get through this whole section in this next part. Alright, we're moving on to environmental factors and how they affect growth, like oxygen, pH. So microbes have to deal with whatever these environmental factors are or adapt to them. Okay, and here are the things we're talking about: temperature, gases, pH, radiation, solutes, all these different things. There's a difference between growth, which is actively replicating and dividing, versus tolerance, which is I'm just surviving but not growing. So we just mentioned that here, but it's not necessarily, I'm not going to tell you which one is growth or tolerance. Sometimes you'll see that when we go through each factor, but we're going to look at the factors and how different microbes might deal with some of these. So we're going to start with temperature. Microbes aren't unable to control their temperature. We can regulate our own temperature, but we're these large multicellular organisms. If you're a microbe, you're just going to be whatever temperature the environment you're in is. Okay, so in that case, what we find is there's a set of cardinal temperatures, which is the range that microbial species can survive in. So what that means, there's going to be a minimum temperature where they're tolerating it. Okay, there's that word, but they're not necessarily growing very well. There's going to be a maximum temperature where they're tolerating it but not doing well. And then in the middle, we're going to have their optimum temperature. That is where they have the best growth. Okay, so we find that, and it's going to be different for your different types of microbes, and that could just depend, for example, for bacteria. The fluidity of their membranes might be affected by heat or cold, but so they can thrive at different temperatures, but it just depends on the chemical makeup of the organism. So this is what I'm saying. In the optimum temperature, this is going to be the best temperature, right? That's the fastest growth rate of growth and metabolism. So for an example, maybe you get sick with some kind of bacteria, and it grows best at your temperature. So at 98.6 degrees Fahrenheit or 37 degrees C, that's the optimum temperature, and it's growing and it's thriving and it's doing great, and you're feeling like crap, and your body finally says enough and starts a fever. Now your temperature goes up. Maybe you're going to push that bacteria out of its optimal range into, it might be at its max, or it might even pass that where it can't survive anymore. At least your white blood cells could take it over. So that's what we're talking about with these range of temperatures. So at optimum, that's the fastest growth rate. That's the most beneficial to that organism. Min, we said, okay, the minimum temperature, that's the lowest temperature. And by the way, this is my shorthand for temperature, where you still have growth and metabolism, okay, but below that, the activities are limited. And I would say this is probably, even at these lower temperatures, I think that's stretching it, saying that there's growth. There's probably not a lot of growth at the very minimum temperatures. Maximum, that's the highest, we said. If the temperature goes above the max, then the growth stops. If it gets even hotter, okay, if it's really high, that's when we see enzymes and proteins start to unravel or denature. That's that word, denature. Just means they come apart. They don't hold their shapes. If you don't have your conformation, you don't have your function. So then they're permanently inactivated, and that would kill the microorganism, which is why fever works. Here's some examples of some ranges. You don't have to memorize these numbers. I thought you just might think this is interesting. But typhus rickettsia, it has a very strict small temperature range. Okay, so it's only going to survive in 32 to 38 degrees Celsius. If you look at a rhinovirus, which is a common cold virus, it does very well in tissue that is below normal physiological temperature. So where do we often get a common cold from is probably in our nose. So it could get into the nose and colonize and then move further into the body. Staphylococcus aureus has a very wide range. There's a reason we have a problem with staph. It grows everywhere. And one of the reasons is, again, the range is from 6 degrees to 46 degrees Celsius. Right? And we can go ahead and describe our microbes kind of based on their temperature preferences. We can put them in these categories, whether they like cold temperatures or warm temperatures. Psychrophiles are cold-loving. Mesophiles are in the middle. And then think heat. Thermophiles like the heat. So let's talk about the cold-loving microbes, the psychrophiles. They like to have an optimum temperature below 15 degrees Celsius. They can grow at 0 degrees Celsius, which is ice. You know, that's when water is freezing. It's cold. They can't grow above 20 degrees C, which would be, to give you an idea, that's about 68 degrees Fahrenheit. Now, there's a category of psychrophiles. It's, I would say it's within the psychrophiles. It's called the psychotrophs. Okay, so this word is different. Trophs. This is phile. This is trophs. They're sometimes called facultative psychrophiles. They can grow in the cold. They're going to grow slowly in the cold, but their optimum is somewhere between 15 to 30 degrees. So they're not necessarily psychrophiles, okay? So they don't like to grow in cold, but they don't die in cold. Okay? They can grow, they can hang out in there, and they're not going to die. Those are not good for us. If we're doing something like freezing something, you think, well, you froze it, you killed it, and then they're hanging out in there and they're fine. Or I should say in the refrigerator, not your freezer, but your refrigerator. You think you're preserving it, but it's hanging out and they're just doing fine. You put it back on the counter and it starts growing like crazy. Okay, the mesophiles, this is most of our medically significant organisms because these are the ones that colonize the human body. So somewhere between 10 and 50 degrees Celsius. Optimum would be 20 to 40 degrees C, which, if to give you an idea on Fahrenheit, 68 degrees up to 104. That's pretty high. So even if you have a fever, you better have a really high fever to get rid of these things. Most of the human pathogens are between 30 and 40 degrees. Okay, thermoduric or thermoduric microbes, these ones survive short exposures to heat. These are not great because they can survive if they do a flash heat, like a pasteurizing food. For example, Clostridium, which causes botulism, can survive that process, and then you can get it in food, and you think it's been processed and it's safe when it actually has a microbe growing in there. The thermophiles. So phile is loving. These are heat loving. They like temperatures above 45 degrees C, which is 113 degrees Fahrenheit. They're usually in soil or water that's around volcanic activity and habitats that are right in the sun. So they range anywhere from 45 to 80 degrees C, which is over 200 degrees Fahrenheit. It's very hot. So here to give you an idea, if we were looking at this on a graph with temperature as our x-axis up to 130 degrees C, and then the rate of growth, you're kind of seeing where the minimum is for an organism is down here. Growth is slow. At the top of the peak, that's their optimum growth because that is the highest rate of growth. So that's going to be the optimum temperature if we were to come down. And for the blue line, that's the psychrophiles. They like cold weather. And you're coming down right around, we'll say, 4 degrees Celsius. And then as we start to get warmer and warmer, that rate of growth is going to drop. That's going to be their maximum temperature. They're not going to grow past 15 in this example. So this is a cool chart showing you the psychrophiles, the psychotrophs, which can survive cold but don't necessarily like it, mesophiles, and they're all overlapping a little. And then here's the thermophiles. And they're going to live in places like thermal hot springs and deep sea hydrothermal vents. It's up to here. Here's the extreme thermophiles. Those are the ones that get really hot. So that was all temperature. Atmospheric gases are also important for the growth of organisms. The big ones are going to be oxygen and carbon dioxide. So we're going to start with oxygen first. That's the bigger one. The reason is oxygen can be toxic. So when we're looking at microbes, we put them into 3 categories for oxygen. They can use oxygen and they detox it. Okay, we have to detox it too. They don't use it, nor do they detoxify it. Okay, so which means that they can't be in oxygen or they would die. And then category 3 is they don't use oxygen, but they can detox it. So they can be in an oxygen environment and not die, but they're not going to use it. So what happens? We know when oxygen is used in cellular reactions, and we're thinking like in your mitochondria when you're doing cellular respiration, this can happen. Several toxic products are formed. So one would be right here. This is the singlet oxygen. It looks like this or like this with a dot on the top. Extremely reactive molecules. Okay, and they're produced sometimes by our own white blood cells, our phagocytes. They help kill an invading microbe. Okay, but if you're on the end, if you are a little microbe and you're exposed to these, they can oxidize membranes and lipid membranes. They can destroy the cell. And so they don't want to be exposed to this. They need a way to detox this singlet oxygen. And the other types of oxygen species that are toxic would be superoxide anion, hydrogen peroxide, and then hydroxyl radicals. And so all of these are all kinds of issues. And again, these are just byproducts of oxygen when we use them in metabolic processes. So the enzymes that are required to detox these, and we do have these, is catalase and superoxide dismutase. There's two pathways for this. This is a two-step kind of detox program. So you need both of these enzymes to be able to process oxygen and not have the toxicity from it. So how does this relate to those categories we were just talking about in the previous slide? Aerobes, or organisms that use oxygen, aerobic organisms, they're using oxygen in their metabolism. They have both of those enzymes so they can process any kind of toxic compounds or byproducts. An obligate aerobe would be one that cannot grow without oxygen. They have to have oxygen. Okay, so obviously they have the enzymes to deal with all the toxic byproducts. A facultative anaerobe. Okay, this would be an aerobe that doesn't require oxygen. Okay, so that means it has all the enzymes to break it down. It doesn't have to have it. So it can grow without oxygen. If oxygen is present, it can use oxygen. It can do aerobic respiration, and it can have those enzymes to break down any toxic byproducts. If there is no oxygen, it's also fine. It can do fermentation, and it's going to be just fine and have an energy source. A microaerophile is just what it sounds like. Aerophile is loving. This is an air-loving, but micro, just a little bit of love, okay, not a lot. It doesn't grow well at a normal atmospheric condition of oxygen. It just needs a little bit of oxygen in its metabolism. So anywhere where there's just a little bit of oxygen, it can do pretty well. This could be in different pockets of soil or in habitats of water. We have areas in our body where some are anaerobic, where we don't have oxygen, like little pockets in our mouth, for example, our intestines. But then there's other areas where we have a small amount of oxygen where a microaerophile could colonize and do well. Not good for us. An anaerobe, they do not have the enzymes to break down those toxic byproducts of oxygen. Oxygen will kill them. So a strict, or you would say an obligate anaerobe, cannot tolerate free oxygen. And that word free is important, okay, because they can live. They live in lakes, they live in oceans, they can live in soil. And what we mean is that free oxygen that's available there, that will actually be harmful for them. But if it's just in the water column, then it's okay. For us, these are the ones that can cause cavities in our gums. They can cause problems in the large intestine where we have these areas with no oxygen. There's also aerotolerant anaerobes. They don't utilize the oxygen. They can survive, they can grow in its presence, but they're not harmed by it. So they can break down what they need to. CO2 we just really mentioned very quickly. All microbes need some carbon dioxide in their metabolism, and that's normal, but a capnophile is one that really loves CO2. And they grow better at a high level of CO2 than would be present normally. And an example might be Neisseria, which causes gonorrhea. So this is important if you were going to isolate this species or you're going to work with it in a culture. In that case, you're going to need to actually do the incubation and work with it in a CO2 incubator, okay, where you have a high level of CO2 from 3 to 10%. Excuse me. So using your basic open hood, or if you're just using a regular fume hood, or whatnot, it's not going to work for these organisms. So you would need to know that in advance, or else they're not going to grow. Okay. pH. pH is really important for us. It's really important for all microorganisms. It's the power of hydrogen. It very simply is just the concentration of hydrogen ions in a solution. And then we express this on a scale from the number 0 to 14, where we are measuring how many hydrogen ions we have. At pure water, it's pH at 7, which means our hydrogen ions are equal to our hydroxide ions that are in solution. However, that balance gets disrupted as we move away from 7. Okay, if we go decrease our number and we're going down towards 0, we have a solution that's become more acidic, more hydrogen ions versus hydroxide ions. And then if we start moving away from 7 towards 14, we get an alkaline solution. And we're going to have less hydrogen ions, more hydroxide. Most microorganisms live at a pH between 6 to 8 because strong acids and the bases can be very damaging to the cells and enzymes, but there's always going to be exceptions to this, of course, and those are going to be the cool microbes. Obligate acidophiles, these ones need acids. Okay? There are certain types of molds and yeast that tolerate acids very well, and they're common to spoiling pickled foods. Pickled foods generally don't get spoiled because they are of a high acidity, but these guys could get in there and still grow and have a good time. Alkalinophiles, these ones live in hot pools or soils that have a lot of basic minerals in there. There's even some bacteria that can decompose urine, okay, and create an alkaline kind of conditions, and they live in that. Well, that would be important to know for a UTI. Osmophiles. So this is a condition of living in a high solute concentration. We were just previously talking in our part 2 of this chapter about tonicity and how isotonic was really important for most organisms, but some could live in a hypotonic environment, but hypertonic conditions are very hard for anybody to live in. Okay, except for some of we mentioned the halophiles. So there are these obligate halophiles, which are salt lovers. Okay, they require high concentrations of salt for growth. They have modifications in their cell walls and membranes that they can bring salt into the cell and deal with these high levels of solutes. If you put them into a hypotonic habitat, they're going to lyse or explode. Okay, they're not going to do well because, because they're not set up to handle that environment. A facultative halophile is resistant to a high salt concentration, even though they don't normally live in there. Okay, so that would be that they have a way to actually get around that temporarily, but they're not going to do well there. Okay, it's not going to kill them. Okay, barophiles have to do with pressure. Just think of like a barometer. An example would be a deep sea microorganism that has a pressure, of something like 1,000 times what atmospheric pressure, and that would crush normal organisms, but they're able to withstand that pressure. And when we bring them up to normal atmospheric pressure, they usually rupture, which is kind of crazy. And then the last environmental factor we're going to discuss is that between organisms. This page is really just a bunch of definitions, so I do want you to know the difference between these definitions and know what they are, but you don't have to know a ton of detail, just basics. What is this? Most organisms are not living by themselves. They're usually multiple organisms around. So let's do non-symbiotic first, because there's not as many. These ones would be, they're free living, which means they don't have to depend on another microorganism for survival. But if they're living together, even though they're non-symbiotic, sometimes they can sort of cooperate and share their nutrients. We would say that's synergism, and we see that, for example, in a biofilm, where maybe one organism is giving off something that the other one needs, or vice versa. So they don't have to live together to survive, but if they're next to one another, they're going to do well. Antagonism is more common. This is going to be, they're competing for nutrients or space. So they're antagonistic to each other. And this is where we get a lot of our antibiotics from. So bacteria and fungi will release these antibiotics into the area around them to try to kill off other microorganisms so they can colonize. And then, of course, we've used that for medicine. Symbiosis, symbiotic relationships. This is when organisms live together in some sort of nutritional relationship, and it's required for one's survival, at least one, and sometimes both. Okay, so someone's going to benefit, and if you don't have each other, you're not going to survive. So mutualism is obligatory. Okay, they're dependent. Both members are going to benefit, okay, and they need each other. Commensal is when the commensal is the benefit, the one that benefits. The other organism isn't harmed, but doesn't necessarily benefit. Okay, so they're together, but that one requires the other. And only one is really having any kind of success. Parasitism, the parasite is dependent. Okay, it can't live without the other organism, but the host in this case is harmed. So here, they both need each other, they're both happy. Here, one is benefiting, the other is not in any way, but it's not harmed. Here, one benefits and the other is harmed. Okay, so that's kind of the basics. And then we have our normal microbiota that, just to mention this, that live on us. We have a lot of different bacteria that live on our skin. They live in our alimentary canal. This is your digestive tract. In different parts of the body, there's microorganisms that live in the vagina. So, and they're important for our health overall. We can see that these things, when they go out of balance, they cause problems for us. For example, if the pH goes off balance on our skin or in the vagina, you can get a yeast infection on the skin or in the vagina from that, because your normal microbiota aren't there doing their normal job. We see problems with this in our intestines. We have E. coli that live in there. They make vitamin K for us. There's a lot of different microbes that are in there doing some fun things for us. We don't have to have them. We can survive without them, but it's a benefit. But what happens when you take your antibiotics? You just wipe them all out. You kill the good guys and the bad guys. And that's unfortunate. That's why probiotics are really big right now. So a probiotic is simply when you take a solution you're eating, microorganisms that are healthy for our gut. They're called gut bacteria. There's also prebiotics. That's a waste of money. Don't buy those. Prebiotics are just sugars that feed the bacteria. Now, you don't need those because your body is essentially giving them all the prebiotics they would need. It's just sugars. So probiotics are the actual bacteria. So if you're going to take an antibiotic, oftentimes they suggest you eat those. You can buy them. Sometimes they're in a food you're already eating. They'll say added probiotics, but the best way, you can just buy straight probiotics at a health food store, but the best way to get them is just naturally in something like yogurt, or there's different, kefir is another type of yogurt drink that just naturally have them. Okay, so next, the last part is going to be 7.3. Let me see where we're at. Okay, we're good. We're going to stop here, and then hopefully we can knock out section 7.3 in the next video.